Hello, welcome everybody. Thank you for joining this IGATech webinar series. Today we are here with Giovanni Giuliano from Enea, and the webinar will be on a new emerging technology, which is single primer extension that can be used for characterization and management of gene banks and pre-breeding plant material. I will first give a technological introduction to the technology, and then I will hand the presentation to Giovanni Giuliano, Research Director at Enea. Just a brief introduction on IGATech. IGATech is a spin-off of the Institute of Applied Genomics in Udine. And the main mission of IGATech is basically to support the research and companies in adopting successfully the latest technologies in the sequencing field. And so one of our major scope is to be always updated with the latest technologies and to work with the highest standard of quality. In order to be compliant in delivering the latest technologies, we are equipped with either platforms and know-how to produce experiments in several fields, ranging from metagenomics to single-cell analysis, and also in agrigenomics fields such as genotyping by sequencing approaches and reconstruction of plant genomes. We have a worldwide collaboration either by our customers or in the framework of research projects, mainly funded by the European Commissions. And these collaborations usually are the major income of our know-how on deploying newest technologies. And also we are authoring tens of publications in high-ranked journals, where you can see that we are also providing successful leveraging of new technology in several fields. If you have any question during this webinar, please use the panel to write down your question and we will be happy to answer. And anyway, there is a public channel that you can find the URL here, where you can reach this webinar on demand when you want. So going to the genotyping field, you know that genotyping is basically a sampling of the diversity within a given population. And there are many applications that would exploit this application, and they can range from trait selection and breeding strategies, or just to analyze the population structure, or find association with phenotypic trait or diseases. And depending on the type of the analysis, the flexibility and the scale of the genotyping is pivotal. We can go from the need of very few markers for phylogeny up to hundreds of thousands of markers for running association studies. And so with the flexibility, we found a very good companion on this SPET technology, which is basically a targeted sequencing strategy, where instead of using the classical capture sequencing by hybridization, here the targeting is driven by single primer extension. So the figure here depicts how it works. Pretending that we have a known SNP, which are not mandatory but can be targeted, locus-specific primers can be designed upstream of the target site, and those primers will trigger the template synthesis of the downstream locus. So the downstream locus will be enriched and sequenced on Illumina platforms. And the sequencing will provide SNP information to either the target site, but also discover and simultaneously genotype other unknown, previously unknown polymorphic sites. And the technology can be adopted on different reading strategies. So you can either use very short reading strategy, like 75 base per reading, just to genotype the target sites, or exploit the full length of Illumina platform and also using parent strategy to discover much more novel SNP sites. So how the procedure works, so basically pretending that we have in this, like in this figure, 384 samples to be genotyped, on a single sample level, there is a fragmentation step, which is followed by the ligation of standard adapters, so universal adapters. Then all those samples are pulled together in a two-tier approach. So basically, first samples are pulled in a plate, and then the whole plate, the whole original plate is pulled within a single tube. And in that single tube, the enrichment happens, followed by the PCR amplification. So that gives you the idea that this technology is very automation friendly and can support a very good economy of scale by reducing to a single tube than the managing of the sequencing template. We have already published a couple of papers on this technology, one where we benchmark the technology with the Illumina Infinium chip, and also one of the first paper provided by the G2P-Sol consortium on the tomato and eggplant germplasm that Giovanni will talk about later. So the main outcome of our seminal paper on maize and black poplar was first that this target genotyping system has an added value because by using non-target SNP site, it is possible to distinguish population structure, like here highlighted by the red circle, which was not very well distinguished by only using target sites. So the ability to always discover new alleles in the population make this system robust to its application in very different gene pools. While on maize, we assess the performance of the technology on a range of different level of coverage saturation. The most indicative is the D figure, where you can see while on homozygous inbred lines, very low coverage is already sufficient to reach the saturation of the accuracy in the call, whilst for hybrids, you need to reach at least a 50x coverage average to ensure the highest reproducibility and recall rate. We are also applying this technology on automation. So we have two systems deployed in our labs. One that is the legacy system, which is semi-automated because we need to run thermocycling off of the deck. We can run out up to 800 samples in a 2-day protocol. Whilst with the new installation of the Tican Dream Prep, we can do everything on the deck. So it's a completely handoff system, which can provide 4 plates genotyping in a 2-day protocol. And we assess the reproducibility of the protocol by comparing libraries that have been produced either by hand or on those automated platforms. Comparing the Allegro target genotyping with array-based platform, the major benefits of Allegro is that firstly, it is leveraging the continuous decrease in NGS cost. There is no platform lock-in because basically it's a technology that can be then applied on any platform which can provide sequencing. It's a very scalable solution because it can range from a panel of 1,000 of loci up to 100,000 of loci. And most importantly, it can always genotype unknown and previously undiscovered new alleles. So therefore, it carries no ascertainment bias by the original germ plants that provide SNP information for its design, which are not mandatory, as I said. You can also design a panel without having any knowledge on existing polymorphism. Also, the technology can not only target single sites, but it can be used to generate a tiling path to fully resequence entire genes that are of major interest for a given crop. This technology has already been applied in 3 European funded projects that we participate or we contribute. And one of the latest is the G2P-Sol project, where more than 20,000 accessions across tomato and eggplant have been genotyped with this technology. And again, Giovanni will be the most knowledgeable person to describe the results of this project. I also want to point your attention that we have a web page on our website where we are listing out the already available panels that we have developed and that can be readily used for other genotyping experiments. And you can see we have already a good selection among fruit and vegetables and forestry, and we are already designing many more to be deployed in the future. Okay, thank you. Now I will hand the presentation to Giovanni Giuliano, which is the research director at ENEA, the Italian Agency for New Technology. Thank you, Giovanni.
Thanks, Davide. I hope you see my screen.
Yes, we are.
Let me get rid of this and let me change my laser pointer. Okay, here we are. So I will be talking to you about the application of single primer enrichment technology for the characterization of gene bank holdings. So let's talk a little bit about plant genetic resources. There are over 4 million accessions of crop plants and their wild relatives in worldwide germ pass banks. And for the majority of such accessions, passport and phenotypic data and even botanical classification are incomplete. So we started the G2P-Sol project in 2016. It ended at the end of 2021. And this project involves 19 partners, which are the dots in red on this map. And there are also some collaborating institutions which are the dots in orange. As you can see, it's a really worldwide project. It includes very large gene banks, such as the World Vegetable Center in Taiwan, SIP in South America, in Peru, and IPK, which is the largest European gene bank, and others as well. And it's devoted to Solanaceae, to the 4 major Solanaceae. So potato, pepper, eggplant, and tomato. And of these 4 crops, which are extremely important, they make up about 66% of the European GDP generated by horticulture. And potato especially is extremely important. It's the staple crop for about 800 million people worldwide, the main source of calories for 800 million people worldwide. In the gene banks of the project, we had about 64,000 accessions. which are about half of those that you find on Genesis, which is the largest compilation of the largest catalog, the largest online catalog of worldwide accessions, so gene banks. These 64,000, about 14,000 are wild relatives, which, as some of you probably know, are very important for introgressing traits, resistances, and others that have been lost during domestication. Most represented one is tomato, followed by potato, pepper, and eggplant is the least represented in this collection. And we look at this collection as like a priceless phenotype library without a comprehensive catalog, because during collection, very often the passport data for these accessions are incomplete. During manual compilation or exchange of accessions between gene banks, there are mix-ups. So the catalog to access the accession, the phenotype that you want, is partially, I would say, strongly incomplete. So the objectives of this project were make an inventory of the existing accessions in the gene banks that participated to the project with the available passport and phenotypic data, then genotype as much as possible with a low density method, about 2,000 markers of these accessions, to really recheck their assignments, their phylogenetic assignments, and their population structures. And then from these, we made core collections, we phenotyped them, and we genotyped them at high density. And finally, we made GWAS, genome-wide association studies on all 4 crops out of all these studies. So it's a stepwise strategy, and I'll be talking mostly today about the second step, which is the low density genotyping. So this is the inventory. I said we had about 64,000 accessions. Of these, we had passport data for 56,000 of them, phenotypic data, which are collected by gene banks during seed multiplication, for 28,000, and images for 17,000. And you can find all these already in a single place, which is the GWSOL gateway here, and it's for everyone to see. The second step was the low density genotyping. So what we did first was to work out 3 robust DNA preparation protocols that were ring tested among the partners, some of them very, very experienced in DNA preparation, some others were less experienced. So we gave them these 3 protocols and we ring tested them, and basically each partner chose the protocol that better suited them and their setup. And then they went on to prepare the DNA from their accessions in 96-well format and shipped them to the genotyping partners, which were ENEA for tomato, the IPK, the German gene bank, for pepper, James Hutton Institute in Scotland for potato, and University of Torino for eggplant. And in these 96-well plates, we included 1 control accession, which is the reference genotype for which there is a reference genome. It was included in well A1, and then I believe well A12 was left empty for some other controls to be added. And then we and UNITO chose, as we will see, SPET for these genotyping. So we performed QC, normalized DNA concentration, and then shipped to IGA for library preparation and sequencing. So initially, when we wrote the project, we thought of using GBS. You all probably know about GBS. It's a reduced representation sequencing protocol in which you restrict with the restriction enzyme, you sequence the sites next to the restriction enzyme, and then you pool the samples and sequence the pools, and you get random SNPs in the stretches of DNA flanking the restriction sites. SPET instead, as David explained very well, is a targeted technology in which you target certain regions of the genome using a single primer. So why did we end up using SPET for tomato and eggplant? The reason was very simple. Keygene, which was holding the patent to GBS, learned about our project, and they approached us and they asked for royalties in order to be able to use the protocol for our project. And the partners who had already GBS in-house working, they chose to go with the GBS and to pay the royalties. The two partners that did not have GBS in-house working, which were us and University of Torino, decided instead to switch technology and start this effort with SPET. And at that time, there were not very many papers published on SPET, so it was a little bit of a risk-taking on our side, which worked, as we will see, very well. So let me summarize the characteristics of the two technologies. GBS is random markers. The throughput is very high. The cost for low density is around the cost that we budgeted in our project for low density was 18 euros per sample. And for high density, we mean 20,000 markers, we budgeted 35 euros per sample. There is no freedom to operate in the sense that if you are a small fish, probably Keygene will not come to you asking for royalties, but if you have a project with 30, 40,000 samples to genotype like we did, then Keygene will contact you and ask for payment of royalties. SPET, on the other hand, is targeted. It has very high throughput. The cost is comparable. So we budgeted the same costs for low and high density for the two technologies. And if you buy the kit, you have freedom to operate. So it's a little bit like Taq polymerase. It was covered by a patent, but then if you bought the licensed Taq polymerase, you were free to use it for PCR. So what are the results of this? We genotyped about 40,000 accessions overall, so we reached the target, about 10,000 for pepper and 5,000 for potato with GBS, around 16,000 for tomato and 3,600 for eggplant with SPET. And also we genotyped a sizable amount of prebreeding lines for, QDL mapping and other reasons. And again, the largest work was done in tomato. And let me give you, compare the two technologies with GBS, with a coverage of about 1 to 2 million reads per individual per sample. We called about 3,000 SNPs for pepper and 13,000 SNPs, high quality SNPs, for potato, which is much more polymorphic. With SPET, we reached significant numbers of SNPs, 6,000 for tomato and about 12,000 for eggplant, with much lower coverage. So SPET is more efficient for per amount of sequencing than GBS is. And that's not surprising because we directed our probes to regions that were polymorphic. So how did we design these panels? Davide gave us a big hand with that. So we took, there were plenty of resequencing data for tomato online, 237 accessions, and we chose them 90% to be Lycopersicum, so the tomato, the cultivated tomato, and 10% to be its closest wild ancestor, which is Solanum pimpinellifolium. And we selected about 14,000 SNPs, one every 2.5 genes. The SNPs were, almost all of them were own genes or introns or 5' UTRs or promoters. We sequenced 24 diverse accessions, and we chose the 5,000 markers that gave intermediate coverage. What happens is that if you have a collection of probes, some will work poorly, so they will give low coverage. Some will work very well, they will give a very high coverage, and then the majority will be just intermediate. If you have too many of this or too many of this, the representation of your SNP panel in the final pool of sequences is unbalanced. So you want to choose in this area here in order to have a balanced representation. So for eggplant, the work was very similar. We had very much fewer resequenced accessions. We had 8 of them made within the consortium. 7 melongena, and 1 is the incanum, which is the related wild species. We selected 12K SNPs, and we redid the whole pipeline, and finally we selected 5,000 for each of these 2. And if you look at the distribution of chromosomes, these are distributed on the chromosome arms, which is not surprising because both tomato and eggplant have very few genes in pericentromeric heterochromatin. So these marker poor regions are also regions that have very few genes and that have very low recombination density. So the markers are on the recombination reach and gene reach arms of the 2 species. So let's talk about robustness. So these are 4 different partners in the project. One of them used a CTAB modified protocol. One used the standard QIAGEN mini-column protocol, and 2 of them used LGC's BDEX, which is a magnetic bead-based protocol. When we sent the normalized DNAs from these 4 partners for sequencing, we got anywhere between 379,000 reads per sample to 700,000 reads per sample. And of these, they mapped in the worst of cases, 89% of the reads mapped. In the best of cases, 94 reads mapped. So you can see that this is a relatively robust protocol across different hands and across different labs and across different protocols. Sometimes a more rough protocol like this gave the best results in terms of mapping, not so good in terms of reads per sample. As David has said, besides the target SNPs, you get novel SNPs that are flanking the target SNP that you chose. And in the populations, we got back not the whole set of 5,000 SNPs. We got less of that, than that. So it means that in our population, some of the chosen SNPs were monomorphic. And we got a large amount, particularly large in eggplant, of novel SNPs that flanked the target SNPs that we based the design on. So again, robustness missing data. We had a little bit more missing data in tomato, 1.18%. This is a percentage comparable to what you get with with the DNA chips. In Melongena the amount of missing data was really very, very low, 0.02%. And as you can see, as you move out from the domesticated species in which the intra-specific SNPs were chosen, you get more and more missing data, and that's understandable because some of the probes will find a polymorphism and they will not hybridize well on a species, on a wild species. But the amount of missing data was always acceptable. So this means that you can use this technology to genotype not a single species, but a complex species that is as far away in evolution as 3.5 million years. And the other good thing about the technology is that if you get if you have a tetraploid species, let's say, you can get all 4 alleles if you have enough coverage. While with DNA chips, you only get 2 alleles if you have a 2-color based technology. Of course, with GBS, you again also GBS is you get all the alleles that you have in polyploids, provided you give a good enough coverage. So these are the maximum likelihood trees of the 15,000 tomato and 3,500 eggplant accessions, and just to show you that all the controls go together, cluster together, both in tomato and in eggplant, and that the species cluster together in maximum likelihood trees, so everything seems to be fine. And here I would like to show you what happens if you use the intra-specific SNPs that were used to make the design, or you use all the SNPs that you discover in the genotyping. So if you use all the SNPs, the inter-specific SNPs prevail, and you can see the species sort of collapse in PCA. So here's Melongena, the purple one, and it's large, very, very collapsed. And then here is, for instance, Macrocarpon, which is another cultivated species. Here is Ethiopicum, which is another cultivated species, both from Africa, Lineanum, etc., etc. If you use the target SNPs, which, as I said, are intra-specific, you can see the eggplant explodes in both components of the PCA, and the wild species sort of collapse in a corner. So depending on what you want to do, you want to do large-scale phylogenetic studies, or you want to discriminate between different accessions of the same species, you can apply one or the other type of SNPs. And this discrimination here goes as far as to distinguish for the eggplant the accessions that come from the Middle East, whose origin is from the Middle East, from those that come from the Indian subcontinent. So it's using the target SNPs, you get a really very fine mapping of your population, genetic mapping of your population. This, as I said, in tomato, this technology has been used also to for the fine mapping of backcross inbred populations from Danny Zamir's lab. He had done backcross inbred populations between Solanum penelliae and a determinate and indeterminate tomato variety. He had 1,500 backcross inbred of this and about 800 of this. and we genotype them for him. And here's the genetic distance versus the physical distance, and you can see that sometimes you get big jumps in the genetic distance in a very short space, and these jumps are recombination hotspots. So you can see here the pericentrometric heterochromatin, very little recombination here. These are the arms. There is a high degree of recombination, but not only that, you can even spot recombination hotspots in which the line becomes almost vertical. Look here, there's a very large recombination hotspot here. There is a new in a new project that started 1 year ago, Harnestom. We are making a new design. So we are going to take the best SNPs from our 5K panel. We are going to take the best SNPs from another widely used platform in the tomato community, which is the Solcap Infinium chip that has 7,000 SNPs, was developed by an American Solcap collaborative project. We will add SNPs, functional SNPs linked to important traits. Then we will filter out low information SNPs like low math, high missing data, and these will result in a new 12K public design, which we believe will be sufficient both for GWAS and fine mapping. The cost will be about 28 euros per genotype for the first 1536 samples. Of course, with increasing volumes, the cost goes down. So the design has started, and if you are interested in joining the consortium, please drop me a line at this email. So the main conclusions are that SPET is a genotyping method that can be used for both targeted genotyping and SNP discovery. The use of single primers, that's what we did in G2P-Sol. Sometimes people target each SNP with 2 different primers, one on the plus and one on the minus strand. In order to maximize our output, we just designed a single primer per SNP, and it worked fine. The use of single primers rather than primer pairs greatly simplifies the assay design. It's robust. It works well with DNAs from different labs and from wild relatives, up to 3.5 million years distant from the species used for the design. And typically, it can be used with 5 to 100,000 probes per assay. And the cost for 100,000 probes per assay is not, it does not scale with number of probes. So it's the cost for the assay is only about 3-fold higher than for 5,000 probes, which means that you can get a relatively a high number of SNPs for a relatively low cost. Of course, the sequencing costs scale linearly, but the cost of the assay and the library preparation does not. And this is the people I wanted to acknowledge, of the G2P-Sol partners, especially the people at Enea and at the University of Torino that did all the normalization and quality controls, and also the people in IGA that did the library preparation and sequencing, and the bioinformatics then were again on us. So we did the bioinformatics, and Stephanie Bruns from Tecan lent a big hand for in designing the assays. The funding came from Horizon 2020, mainly G2P-Sol, but there is also funding now from the Harnestom project. And thank you very much for the attention. I'll take any questions you have.
Thank you very much, Giovanni. We actually stayed in 40 minutes sharp. So I have here already a list of questions that were raised during the presentation. The first one which I would take is on how the technology compare to the sequence capture in terms of cost and flexibility. I mean, the answer can be long, but to make it short, sequence capture has usually a quite high upfront investment in terms of probe designs, and usually is more favorable if you like to resequence large region of the genomes. Let's say just like the exome or a large panel of genes. The efficiency of sequence capture is usually low. I mean, depending on the level of the optimization of the panel, you can range from 50% to 70%. SPET, on that sense, is performing better because the percentage of on-target reads is usually 90%. So there are very little sequencing wasted on region that you don't want to target. So I think that also the cost of labor and preparation reagents is more expensive on sequence capture, because in sequence capture usually you pull, depending on how much input DNA, you can go from 8, 24 up to 24 samples per enrichment, and the enrichment is quite sensitive procedure. Here, as I showed, you can easily work on a single tube, having 96 samples in that single tube. So I think that the long story short is that SPET can provide a higher degree of flexibility, and also it's more beneficial in terms of per sample cost. Like Giovanni mentioned, you see the cost per sample is about tens of euro per sample, usually with standard sequencing capture enrichment strategies. You are more on the 100 per sample. The second question is how other polymorphisms can impact the design of probes. This is a very good question, because actually this is something that can be taken into account during the design, because those using SPET technology are primers that are on average 40 base per long. So what you don't want to have is polymorphism in the very 10 bases at the 3' end of the primer that might disrupt the annealing and the polymerase extension. So usually when other sequencing resources are available, we leverage those to make a SNP call and instruct the design to not land on those polymorphic sites to avoid allele-specific dropouts. Of course, if there are major structural variations that are not explored during the whole genome sequencing, those may cause a drop in some specific alleles, but those have been shown to be minor. And I have a question that is related on what's the best design strategy that can be applied to select target SNPs or regions. So I think I can hand this question to Giovanni.
Yeah, so the best strategy is, in my view, to select intra-specific SNPs, because as we saw, especially for crops or species with low genetic basis, those are the most difficult to find. When you have enough intra-specific SNPs, then in the flanking regions, you will certainly find inter-specific ones. So that's why we started with intra-specific in genes, in regions that have functional significance, and that in the 2 crops we studied have also high recombination rates. So you want to direct your SNPs if you want to do, let's say, GWAS studies or any other studying recombination, you want to have your probes in regions that are not in large linkage disequilibrium with each other. So those are the two. And third criterion is high information content of the SNP. Of course, if there is a SNP that is present only in 1% of the accessions in your population, you are going to get much less information, much less discriminating power than for a probe, a SNP that is present in around 50% of your accessions. So high math, intraspecific, and on genes in regions that have high recombination frequency. That's what we chose to do, and it worked pretty well.
Okay, thank you, Giovanni. The last question is about what is the minimum requirement of DNA to be processed? I can answer this one by just saying that usually we work happily with anything around 100 nanogram per sample, but we also been able to work with just 50 nanogram per sample. So the quantity is not that much demanding. It can be easily achieved with all the standard extraction protocol, even the most high throughput and low yield. I think that the only thing that might need to be taken into account is to provide DNAs that are homogeneous in quantity and quality, because as it has been shown, it's a pool strategy. So you pretend to start with the same amount of DNA for each sample within the plate and then pool before the enrichment step. So if you have a comparable amount of DNA quantity across samples, that will result in comparable amount of sequencing yields across samples. Instead, if we have too high variability in terms of quantity or quality, that will correlate with the sequencing yield, eventually providing more missing data point for some samples. So usually the quantity is something that can be managed by just running fluorimetric-based quantification and then normalization of the DNA accordingly. For the quality, it comes with an homogeneous sampling method. So to sample DNA from the same tissue type and development stage eventually, and of course to use the same extraction method across the plate to avoid the big discrepancy in quality caused by the extraction method or secondary metabolite leftovers.
Yes, in our case, all the extractions were from 3 weeks old seedlings, single seedlings. So we chose a strategy not to pool samples, to take a single seedling per accession, which has its advantages and its drawbacks, because you sort of miss heterogeneous accessions this way, but you maximize the amount of samples you can do. And yes, we tried as much as possible to have in the same plate samples from a single partner, done with a single method. And of course, there was this robotic normalization. So we read the whole plate with multiplate reader, fluorescent picogreen and multiplate reader. And then for plates in which there were many bad samples, we did cherry picking. So we consolidated the plates into a plate with all the samples, with all good samples. And anyhow, we did all this normalization. So robotics normalization with spreadsheets coming out from the plate reader. So we fed them to the robot, and then the robot diluted all the samples to the same concentration. And finally, we did random electrophoresis of, let's say, 1 column per plate to have a look outside the quality of the DNA. Was it very degraded? Was it in good shape, etc., etc.? So all these tedious work succeeded, ended up in a very high success rate. So we had a failure rate about 5%, which is very good.
Okay. Thank you, Giovanni. I think there are no more questions at this time, so I think we can close the session. Again, thank you, Giovanni, for your very precious contribution to this webinar series. Let me just recall that this webinar in the following days will be available on demand from our website. As well, on our website, you will find links to the seminal papers that we already published on this technology and other specification regarding sample requirements. Thank you to all of you for participating to this webinar. See you soon on the next one. Thank you.
Thank you. Bye-bye.
Thank you.